Hello, my name is Elli Brimble with Citizen, and today I'm going to be sharing results with you from our Map It Forward study, which is looking to characterize real-world patterns of oncogenomic testing in cholangiocarcinoma. These are my conflicts of interest. I am employed by and have stock options with Citizen. And I'm going to start today by sharing an overview of the Citizen platform. Citizen is a patient-mediated platform that collects and stores medical records and imaging on behalf of patients. We have also developed a machine learning and natural language processing assisted approach to extraction, and that data is also reviewed by clinicians as part of our QA and QC. The resulting output of our data is a research-ready dataset that is structured and normalized. As we collect information across many different institutions, it's important that we have a systematic approach to capturing data. We think about this in two ways. At the top level, we have our disease model, which essentially serves as the conceptual foundation for our products. The disease model defines what types of data we extract and ensures that those data elements are relevant to both clinical and research indications. And the model also establishes relationships between those data elements. So in a way, we're generating a network of data rather than something that's linear. Within the disease model, we also have an ontology, which we can think of as like a expertly curated dictionary. This dictionary is made up of terms from internationally recognized terminologies, and it's critical to support harmonization and normalization of our data. It also allows for us to integrate our data seamlessly with other platforms. Another important thing for us to consider is how we can address data gaps. And we're lucky in that we are a patient-centric model, so much of this is already accounted for. Some of the things that we consider are that we're not limited to a single institution or EMR, and we're actually able to collect records across many different providers, including commercial and academic labs, which you can imagine is really important for a study around genetic information. We also maintain contact with participants, and so we're able to reach out to them to request updated information or to request missing pieces of information. We've also built internal processes that flag when key documents are missing and trigger a collection flow to be able to collect that information in a somewhat automated fashion. As a result, we're able to collect 99.9% of collectible records, and only a small amount are deemed to be not collectible because they do not exist. So I'd like to share with you more now about the study that we have developed in partnership with the Cholangiocarcinoma Foundation. Our primary objective is to describe and characterize real-world patterns of oncogenomic testing in cholangiocarcinoma. As I've shared, Citizen maintains a data bank to collect and store data that's been extracted from participants' medical records. Users of the platform can also consent to share their de-identified data for research purposes. In order to evaluate our primary objective, Citizen has obtained an IRB determination of exemption. And for the purposes of this study, we're defining oncogenomics as any germline or somatic testing that's been ordered as part of a participant's routine clinical care. Our recruitment for the study is ongoing, but at the time of analysis, we had completed data extraction for 153 participants, and of those, 133 have consented to participate in research. So this shows that we have a very highly engaged community dedicated to research. Once we have completed the extraction, we're able to identify those profiles that appear to have no information around oncogenomic testing. And so we actually sent a survey out to those individuals to clarify whether or not that absence of information is because they hadn't had any testing performed, or perhaps because we may have missed it as part of our collection. We had a response rate of about 37%, which is not bad considering this was an unpaid survey. And within that, we had a subset that confirmed that they had not had any testing. And we also had 8 individuals who were able to place new requests for records that we went out to collect. So in total, of the 133 participants, 94 had any oncogenomic testing. In the graph here on the right, I am showing the distribution of age of participants. The average age was 60.7 years, and it ranged from 22 to 88 years. And you can see that most individuals are clustering around 51 to 70 years of age. The majority of participants in the study were female. This is a little bit of a busy slide, so I'm going to direct your attention to the first graph here on the left. This is another histogram, but showing this time age at diagnosis. And so again, we're seeing that most people are being diagnosed between 51 and 70 years of age. But in this case, the range of diagnoses was also 22 to 88 years. And the average age was 57.9 years. We looked to see whether age at diagnosis was different between males and females, but we did not see any difference in this case. If we look now at the middle graph, this is looking at the number of diagnoses per year, and we see that more than half of our participants were diagnosed recently within the past 2 years. And this we know is consistent with the high degree of morbidity and mortality associated with cholangiocarcinoma. When we looked at the distribution of stage in the population, we saw again that almost 3/4 were diagnosed with advanced or metastatic disease, again, consistent with a known pathogenesis of cholangiocarcinoma. All right, switching gears, I'm going to dive in now to some of the data we pulled around oncogenomic testing. So the first pie chart we're looking at here on the left-hand side is showing the frequency of genetic testing ordered from different commercial or academic labs. And when you look here at the legend, I'm sure you'll see a number of familiar names, but somewhat impressively, we see that almost half of tests ordered in this population were from 2 commercial labs alone. If we look now at the second pie chart over here, this is looking at the frequency of different types of testing. So in this medium colored box here, we're looking at the participants who had germline testing only. This was only about 7.5%. We can see that the majority here, around 69%, had somatic or tumor testing only, and about 1/4 of participants had both germline and somatic testing. As I discussed in an earlier slide, about 71% of participants had any oncogenomic testing at all, and the average number of tests per participants was 1.6, but this ranged from 1 to 6. And interestingly, I did want to highlight that of those individuals who had any germline testing, we did identify a positive result in one, and this was in the gene MLH1 associated with Lynch syndrome. So there is certainly yield to both germline and somatic testing in this population. We also wanted to see if we could identify any factors that contributed to uptake of oncogenomic testing. And so the first graph here is looking at age at diagnosis, and the first bar is individuals who did not receive any oncogenomic testing, and the second one is those who did. And we see a very small but significant age or difference in age at diagnosis, where those individuals who received testing were diagnosed at a younger age than those who were not. We also looked at the distribution of stage at diagnosis and the percent of individuals who had any oncogenomic testing, but we can see that this is quite variable across stages and didn't necessarily correlate with a later stage or an earlier stage at diagnosis. The next couple of slides are going to talk about the genes in which mutations were commonly found in this group of patients. In the chart here, we're looking at the most common genes where mutations were identified, and we can see right away that we are showing similar patterns that have been previously published in the literature in cholangiocarcinoma. In particular, we know that TP53, IDH1, ARID1A, and FGFR2 are pretty commonly identified in cholangiocarcinoma. Looking specifically at IDH1 variants, we know that there is a specific hotspot mutation in IDH1 that preferentially affects the arginine residue at position 132. And similar to what has been previously reported in cholangiocarcinoma, we also see the common R132C in our population, representing more than half of IDH1 variants, but we do observe some other R132 variants as well. FGFR2 fusions are also commonly reported in cholangiocarcinoma. And again, the common BIC1 fusion partner was present in half of our individuals with an FGFR2 fusion. And then we also know that tumor mutation burden and microsatellite instability are commonly assessed in cholangiocarcinoma, in particular to evaluate the utility of immune checkpoint inhibitors. And in our cohort, the majority of individuals had low tumor mutation burden, and in this case, all tumors were microsatellite stable. All right, so we also wanted to take a look at whether or not the patterns we identified were specific to certain classifications of cholangiocarcinoma. So the top pie chart here is showing the frequency of intrahepatic and extrahepatic cholangiocarcinoma, but you'll notice that a good proportion, around a third of individuals, were not clarified in their medical records. So they did not specify a specific subtype of cholangiocarcinoma. We also looked at the frequency of oncogenomic testing across these three classifications, and we did not observe any significant difference, although we can see that the highest percentage was in intrahepatic cholangiocarcinoma. This heat map here is showing those same genes where we saw most commonly mutated, but instead we're showing the frequency in intrahepatic cholangiocarcinoma and extrahepatic cholangiocarcinoma. In this case, the lighter colors represent a lower frequency and the darker blue represent a higher frequency. And what's interesting here is that we can see a subset of genes that have a darker color, meaning they were more commonly mutated in intrahepatic cholangiocarcinoma and less commonly mutated in extrahepatic cholangiocarcinoma. And then a subset of genes that were more variable. So they were present in both intra and extrahepatic cholangiocarcinoma. And this pattern has actually been previously demonstrated, showing that within our cohort with commercial testing, we're able to recapitulate these established mutational landscapes for the different classifications. And then the last piece of data that I wanted to share with you is looking at pathway analysis. So we took the most commonly mutated genes in the cohort, so any gene that was observed in at least 3 individuals, and we used Reactome to determine whether any pathways were overrepresented by that group of genes. And so here, the overrepresented pathways are in yellow, and they're being mapped to specific groups or biological pathways. And I apologize that this image is a little bit small, but we can start to see some pretty consistent patterns emerging. So we see things like signal transduction, gene expression, disease, immune system, cell cycle, programmed cell death. These are all pathways that have been previously emphasized in cholangiocarcinoma. So in conclusion, Citizen is a patient-mediated platform that supports rich data aggregation and analysis across relevant entities. And by exploring real-world oncogenomic testing patterns, we've identified high diagnostic yields for actionable findings in our population. As I've mentioned, this is an ongoing study, so we are currently recruiting and targeting a sample population of 250 to 300 individuals. And our next steps are going to be to evaluate what impact these actionable findings have on individuals' management and also disease progression. So with that, I'd like to thank the Cholangiocarcinoma Foundation, as well as all of the participants who have shared their data for research. And here is my contact information. Thank you.